Hello, micro students. I wanted to make a second video here. We've been spending time looking at all these macromolecules that are involved in the chemistry of life, right? As we look at what is life made of? Well, we know it's made of cells. Well, what are cells made of? Well, we talk about organelles, Golgi apparatuses and mitochondria and things like that. Well, what are those things made of? Well, that's really what our conversation's been about. And what really everything inside a cell is made of, we've boiled down to four things: proteins, carbohydrates, lipids, and nucleic acids. And we talked about all of those in a previous video. In this one, we want to look at proteins a little bit further. This is where I ended, and I thought the video was getting a little bit long. And so I wanted to make a sort of a second one here for your lesson towards the end of the week to look at um, proteins a little bit. So we had talked about proteins and said, gosh, they do just about everything. That's kind of where we left off, saying functions, I mean, you name it, proteins can do it. If anything happens inside a cell, a protein is the one doing the work. And we said they're really just a chain of these little small beads on the string called amino acids, right? Those are their building blocks. And so we had spent time looking at that. We also talked about protein structure. We said that proteins only function when they're folded up, and they fold up based on which of these amino acids are present and in which order. Okay, that'll determine how it folds, and that fold determines its function. No different than when you cut a key. How you cut that key determines what doors it can open. Okay, and we also talked about this concept, which is actually really important. Enzymes are supposed to be folded up. That's when they're functional. Now, if you apply either heat or chemicals, you can take that folded up protein and unravel it back into a strand like you see over here. We call that denaturation, right? We say that protein has been denatured, and a denatured protein is a useless protein at that point. And so we talk about what we're going to talk about next. Keep that idea of heat and chemicals in mind, okay? We want to look at enzymes for a second. Enzymes are an incredibly important type of protein. And what enzymes do is they're going to speed up chemical reactions inside of cells so that whatever was going to happen happens much faster and in a timely manner. Okay. Well, how do enzymes speed up reactions? Well, this is what they do. I always think of it, and you've probably seen these graphs over and over again through biology and chemistry. I always think about rolling a boulder up a hill. Okay. I imagine if I were going to roll this boulder up this hill, let's say there I'm trying to we'll put a car or something over here that I want to smash on the other side of this boulder, right? I have to do some work to get the boulder up to the top of this hill, and eventually gravity will take over and it will smash this car down here on the other side, right? We call this work that I have to put in, right? This amount of work, that's called activation energy. And you can see that discussed right here. So essentially, you're looking at where are we starting, right, to where we finish, or at the top of the mountain, so to speak. That is our activation energy, the amount of energy we have to put in to get something to happen. Okay. Now, what enzymes do is they're going to lower that activation energy. So instead of having to roll that boulder all the way up the hill in this case, now I only have to get it to here, right? It's essentially bulldozing this hill. It basically eliminates the hill. That way, instead of having to push it all the way up here, I only have to get it to here. And if I can get it to here, gravity will take over and do the job that I want it to do. Okay. Notice our hill is a lot less, therefore, less activation energy. And you can kind of see the comparison between the two here. Okay. And so we say that enzymes, they lower activation energy. That's really what they do. And what happens when you lower the activation energy? Well, it speeds up reactions. It makes things happen faster. And so, no different than, if you have a key, right? A key to get in your house, that key is going to allow you to get in your house pretty quickly, right? If you don't have a key, can you get in your house? Yeah, but you're probably going to have to break a window or do something like that to get in. And so these enzymes are essentially keys. They allow you to get into something, right? They allow you to access something much faster, break something down, whatever you're going to do inside of a cell. And so these enzymes are going to speed up reactions. Okay. And the things that affect how well these enzymes work are really three things: the pH, the temperature, and then how much stuff you're going to be trying to break down and how many enzymes you have. Okay. And every enzyme has sort of a different preference here. And you're going to be looking at an article um, with ethanol, in the process of making ethanol, and looking at sort of the preferences there, right? The pH, really the chemistry that you have that enzyme in. Some enzymes like a neutral pH of 7. The enzymes in your stomach like it at about a 1, 1.5 to 2. These enzymes prefer a more acidic environment. Okay. And then temperature, right? A lot of enzymes in our body function best at 37 degrees Celsius, right? That's our human body temperature. You get them warmer than that or colder than that, they really don't work as well. And one of the things that's happening, and why aren't those enzymes working? Well, enzymes are proteins. And we just learned that if you apply too much heat or chemicals, you can take this functional protein and you can unravel it and turn it into a useless protein. So by applying heat, you can unwind that protein. And that's a problem. That protein isn't going to function anymore. That enzyme won't work. It's like having a key that doesn't work. It's changing the shape of that key. Okay. And so we see this happen. This is why getting a fever is such a big deal. We have enzymes in our body that function really well around 37. You get them warmer than that, they start to denature. And when those enzymes denature, then they don't function as well. Your body starts shutting down. Things can go south pretty quick. Okay. And so if we were to look at a couple enzymes here, just to read some enzyme graphs, I think it's helpful to see these. So we have enzyme A and enzyme B. You'll notice that right here, this is where those enzymes have peak performance, right? Temperature wise, we're looking at temperature and then rate of reaction. Essentially, at what temperature do they operate the best at? And so you can follow it down. Enzyme A operating best at around 37, enzyme B at around 78-ish, right? So which enzyme works best at lower temperatures? Well, obviously enzyme A. At what temp do they both have the same activity? They intersect here around maybe, I don't know, 48 degrees Celsius, and which would be most likely found in the body? Probably enzyme A. Remember, this is Celsius, so 37 degrees Celsius is human body temp, which is about where enzyme A peaks at. But the thing I really want you to pay attention to here is what happens when you get outside of that optimum temperature range, right? You kind of see this curve that goes like this. This enzyme works great here, but as you get cooler or warmer, that activity drops off tremendously, right? I mean, you start looking past, you get enzyme A past about, oh, I don't know, 48 degrees. I mean, it just drops off completely and it doesn't even work at all after about 48 degrees. Okay. So changing a few degrees can cause this to go quite differently, right? You can change that enzyme activity quite a bit. Okay. And so I always call enzymes divas. You'll hear me talk about enzymes being divas. They have to have it just right. And it's kind of like Goldilocks, not too hot, not too cold, just right. They're very picky. They work great, but they're very picky in the body. And we see that in cells as well. And so things that we can do in microbiology to have an impact on our microorganisms is simply changing the temperature. Sometimes that will cause them to grow faster, slower, to change how they do things. You're going to see that temperature is one of those things that we can change, and it can have a big effect on our results. And so I want to make sure we talk about that. And you hear about people like, why is it a problem if you get too cold? What is hypothermia? Things like that. What is heat exhaustion? Really, it's your enzymes. That's what it comes down to. You get them out of their sort of narrow preference, and things start to go bad really quick. And so, be thinking about that. Enzymes are really what control all the chemical reactions in your body. If you don't give them the right environment, they're not going to do their job for you, and your body starts to shut down. Okay. Let's spend a little time looking at how they work. We said enzymes are great at speeding up reactions. So let's take this sucrose, for example. It's a disaccharide. It's a sugar. All right. It's table sugar. It's the stuff you put on your cereal. And sucrose, right, is a disaccharide. It's not usable in this form. It has to be broken down into two monosaccharides, two individual hexagons, right? Oh, forgive my hexagons. Okay. Has to get broken down. Well, you give things enough time. Sometimes they'll break down, sometimes they won't. But we need to speed this up. We have to split these apart much, much quicker. Enzymes will do that for us. And so we have this enzyme, it's called sucrase. Notice it has this special notch in it. That's called your active site. Okay. And that active site is designed to fit perfectly with that substrate or that thing you're trying to break down. It's sort of that lock and key system, okay? And so the first thing is you have this enzyme, it's got an open active site. It's going to bind with your substrate or the thing you're trying to break down. And with the help of a little bit of water, okay, that enzyme is going to break down that disaccharide sucrose into two monosaccharides, glucose and fructose, and then it will release them. There they go. And then those can be used by the cell to go, get energy, those kinds of things, okay? And then your enzyme goes back and is free to go grab another sucrose to get broken down again. You'll notice that enzymes typically end in ACE, ASE, and the first part usually tells you what it's breaking down. So the enzyme sucrase breaks down sucrose. And so you can oftentimes see what the enzyme is or what it does. It's based in its name oftentimes, okay? And so make sure you understand in general how enzymes actually work and what they're doing. We know they lower the hill, right? They make reactions happen faster. They lower the activation energy. They'll break things down quicker through this process that you're seeing here. Okay. Why is it important for us to learn about enzymes and microbiology? They're really what allow living things to function and survive, right? We said anything that has to happen in a cell, a protein needs to do it, and specifically an enzyme protein. Okay. Enzymes are behind the scenes making everything work, making everything happen. Okay. And, it sounds cliche, but really, enzymes make the impossible possible. There's enzymes in laundry detergent. I mean, think of anything that you can think of. Enzymes usually play some sort of role in that process. Okay. Whether it's breaking down food for us or, making new substances that might be useful to us, enzymes are everywhere. Okay. And so I always like to have a picture of this, right? You see this log. We don't typically think of that as food. You don't look at a log and say that looks tasty, right? But it's only because we don't have the right enzymes to break it down. If you had the right enzymes to break down that log, you could extract nutrients from it, and microorganisms can. And so microbes will break that down and use it to get energy, okay? And so that really is the difference when you start looking at the world, the enzymes you have really dictate what you can eat, what you can get nutrients out of, those kinds of things. Some of them, especially in microorganisms we look at, we have endoenzymes. These are ones found inside the cell. And so, as you bring something in the cell, you want to break it down. You have endoenzymes inside the cell to break those things down. You also have exoenzymes. So, if there's something, let's say you're a microorganism and you see this nice big tasty log and you want to break it down, you'll have to release enzymes outside of your cells to help break that log down and eventually extract those smaller nutrients into your cells. Okay. We already talked about enzymes often end in ACE. Okay. And these are just a few on our list here of enzymes that you might run into or important enzymes. DNA polymerase, this is the enzyme that builds DNA for cells. Okay. There's an enzyme zymase. This is the enzyme that is in yeast that actually is able to ferment sugar into alcohol. So whether you're making, beer, wine, whiskey, ethanol for cars, whatever it is, this enzyme, zymase, is one that converts sugar into alcohol. And so, that's a huge industry there. The enzyme PETase, it allows bacteria to break plastic down into food. There's a lot of research on PETase and helping break down plastic waste to make to help break down plastic then in landfills and things like that. Cellulase can break down plant cell walls and plant fibers. That's an important one. We'll see that in the article you're going to look at. Benzyl succinase, or benzyl succinate synthase is its name. This is an enzyme that allows bacteria and other microbes to break down crude oil into food. We don't think of crude oil as a food source, but there are things that can eat it. Why? They have the right enzymes to be able to do that. IgA protease. This is actually a really interesting enzyme if you're interested in like the immune system and illness and things like that. Bacteria have evolved and produced this enzyme and allows them to break down antibodies. So when your immune system is trying to release antibodies to help flag these bacteria, so your white blood cells kill them, bacteria is able to release an enzyme to break down your antibodies so that your white blood cells can't go kill those bacteria cells, kind of a little defense from those bacteria. Lactase, this is a simple one. It allows us to be able to break down lactose sugar in dairy products. So if you do not have the enzyme lactase, you can't process this sugar called lactose, and it can make you sick. And so, if you know somebody who's lactose intolerant, they're just lacking the enzyme lactase. Okay. Catalase is another enzyme. Every living thing that uses oxygen has catalase. It breaks down peroxide into water and oxygen. We'll talk about that later. Amylase is one thing that helps break polysaccharides down into monosaccharides. When you eat pasta, amylase will go to work and turn that into a usable sugar like monosaccharides. And then some of the interesting ones like hyaluronidase. It breaks down hyaluronic acid. So that's a big one right now in skincare. People are using hyaluronic acid as one of their skincare products. And hyaluronic acid basically helps. It's a fluid that goes in between your cells and sort of helps give them structure, gives your skin sort of a tightness and a fullness to it. And so sometimes people will use it for anti-wrinkle and just kind of help with your skin care. This hyaluronidase will actually break that down in your skin, and so it can cause your skin damage. It's found in bacteria. This is one thing that can cause skin damage. And there's also some nastier ones. There's a necrotizing enzyme that will basically break down your cells. This enzyme will destroy your cells and break them down. So you can see this young man here. He must have gotten an injury, poked with something, had an injury. There must have been some bacteria in there. And over time, by releasing this necrotizing enzyme, they're essentially eating away his thumb. You can see a lot of swelling. You definitely see the injury here. Any sort of blackness here is dead tissue. You can see that it's starting to die, and you can see his entire arm is turning red. That's a huge sign of pretty significant infection there. And so, he's at the doctor, hopefully getting it looked at. But, again, necrotizing enzyme. Why can a bacteria do that? It literally comes down to an enzyme it possesses. And then streptolysin is another enzyme here. And it'll break down blood cells. And so oftentimes pathogens will have this streptolysin in them. You can see like in this one, this is a petri dish that's supposed to be red. It has blood cells in it. And you can see these bacteria here, this little streak has broken down all the blood cells and turned the petri dish yellow. Means that you've broken down the blood cells. And so typically if you can break down blood cells, it's not something you'd want in your body. And so they can do this test and look for what's called hemolysis, whether it's beta hemolysis, alpha hemolysis, or gamma hemolysis here. But those are some enzymes. I really wanted to devote a day to this, or at least a little video lecture. Enzymes are behind the scenes causing everything to happen. And so as we look at bacteria and we grow them and we look at why is this bacteria able to do this? Why does this one behave this way? We really need to come back to what enzymes do they possess that would allow them to do that. And that's really going to be our ID to figuring these things out. Okay. So I wanted to talk about those and just make sure we have some insight on enzymes there.